Hello friends, myself Professor Shantaya Gurmut, Department of Botany, MGVC Arts, Commerce and Science College, Madhya Bihar, Karnataka State. Friends, today's class we discuss about plant hormones. In this plant hormone, the example is cytokinin. Cytokinin is a growth promoting hormone. Cytokinins are a class of plant hormones that promote cell divisions or cytokinins in plant roots and shoots. They are involved primarily in cell growth and differentiation, but also affect apical dominance, axillary bud growth, and leaf senescence. There are two types of cytokinins: adenine type cytokinin represented by kinetin, zeatin, and 6 benzyl aminopurine, and phenyl urea type cytokinins like D-phenylurea and thidiurano, that is TDZ. Then chemical means the chemistry of cytokinins. Cytokinins, whether naturally occurring or synthetic, have some structural similarities. They are also derivatives of adenine, which is an purine base of nucleic acids. Secondly, hydrogen atom on the N6 position of adenine ring is substituted by either benzyl resulting in benzyl adenine or by forming kinetin group of zeatin. Occurrence: cytokinins occur freely in plants. In addition, these are also present in the transfer RNA, means tRNA of cells. These occur widely in practically every part of the plant: apical meristem, developing seeds, root nodules, bacterial tumors, and all regions which show active cell division contains cytokinins. The idea of specific substance required for cell division to occur in plants actually dates back to the German physiologist J. Weisner, who in 1892 proposed that initiation of cell division is evoked by endogenous factors, indeed a proper balance among endogenous factors. Somewhat later, the Austrian plant physiologist G. Hamberland reported in 1913 that the unknown substance diffuses from the phloem tissue, which can induce cell division in parenchymatic tissue of potato tubers. In 1941, Johnson's van Orbeek found that the milky endosperm of immature coconut also had this factor, which stimulated cell division and differentiation in every young datura embryos. Jablonski and Skoog in the year 1954 extended the work of Haberland and reported that a substance present in the vascular tissue was responsible for causing cell division in the pith cells. Miller and his co-workers in the year 1954 isolated and purified the cell division substance in a crystallized form and autoclaved herring fish sperm DNA. This active compound was named as kinetin because its ability to promote cell division, and was the first cytokinin to be named. Kinetin was later identified to be 6 furfuryl aminopurine. Later on, the generic name kinetin was suggested to include kinetin and other substances having similar properties. The first naturally occurring cytokinin was isolated and crystallized simultaneously by Miller and D.S. Latham in the year 1963 to 65 from the milky endosperm of corn, that Gia maize, and named Giatin. Latham in the year 1963 proposed the term cytokinins for such substances. Cytokinins are involved in many plant processes, including cell division and shoot and root morphogenesis. They are known to regulate axillary bud growth and apical dominance. According to the direct inhibition hypothesis, these effects result from the ratio of cytokinins to auxin. This theory states that auxin from apical buds travel down shoots to inhibit axillary bud growth. This promotes shoot growth and restricts lateral branching. Cytokinins moves from the roots into the shoots, eventually signalizing later bud growth. Simple experiments support this theory. When the apical bud is removed, the axillary buds are inhibited, lateral growth increases, and plants become bushier. Applying auxins to the cut stem again inhibits the lateral dominance. Moreover, it has been shown that cytokinins alone has no effect on parenchyma cells. When cultured with auxin but no cytokinins, they grow large but do not divide. When cytokinins and auxins are both added together, the cells expand and differentiate. When cytokinin and auxin are present in equal levels, the parenchyma cells from an undifferentiated callus. A higher ratio of cytokinin induces growth of shoot buds, while a higher ratio of auxin induces root formation. Cytokinin signaling in plants is mediated by a two-component phosphorylase. This pathway is initiated by cytokinin binding to a histidine kinase receptor in the endoplasmic reticulum membrane. This results in the autophosphorylation of the receptor, with the phosphate then being transferred to a phosphotransfer protein. The phosphotransfer protein can then phosphorylate. The type B response regulates RR, which are a family of transcription factors. The phosphorylated and thus activated type B, that is RRS, regulate the transcription of numerous genes, including the type A RRS. The type of ARS negatively regulate the pathway. Then we discuss about the practical applications of cytokinins. Number one is cell division, cell enlargement, morphogenesis, counter reaction of apical dominance, breaking of dormancy. First one is cell division. Of all the growth hormones, cytokinins have been found to be a true cell division factor in a number of lower and higher plants, while a number of tissues from meristematic regions are capable of multiplication in the absence of cytokinins auxins. Second one is cell enlargement. A number of workers have found evidences to prove the cytokinins are equally good in causing cell expansions, like auxins and the gibberellins. Then we discuss about morphogenesis. One of the most interesting effects of cytokinins is the phenomenon of organ formation in activity of tissue cultures. Fourth one is counteraction of apical dominance. Exogenous application of cytokinins has been found counteract the usual dominance of the apical bud. The last one is breaking of dormancy. Cytokinins have been demonstrated to be effective in breaking the dormancy of seeds and some other plant organs. They not only break the dormancy but also promote the germination of seeds. Okay, this is about the cytokinins. Thank you friends.